everybody. Whoa, hello. Welcome to Biology Trivia Night. How are all of you doing? So glad to see that you have joined me on this lovely Friday night. At least it is nighttime where I am at for a little bit of glorified biology lessons disguised as a fun game. Hello, Katie Cat. Hello, Mobo. Hello, The Randomer. And Marlon. Hello, TF2 Gamer. So glad you can make it. 44 CPU 7. Welcome. Of course, Icosahedron and Architelos. Please let me know if I've said your screen names correctly. Thank you again for showing up and allowing me to talk about biology disguised as something more fun. Yeah, so today was a great day for me. It was actually the first day of the class that I teach for university. So I teach a university biology class, and it's so crazy to me because I forget that this happens every semester. At the stage that I get students, you know, some of them maybe haven't even had that much experience with pipetting, but by the time they leave, they will have grown by so much, but then they kind of leave forever and I never get to hear about them. So this is a semi-regular reminder that if you've had teachers that were important in your lives, that you make sure that you just occasionally give them updates about how you're doing. If you're worried that they have forgotten about who you are, one, they probably haven't, and two, if they did, they can always check their attendance rules. So if you've had important teachers, please make sure that you let them know. Especially if they are K through 12, because we know that they aren't rich monetarily, so you can definitely try to make their life rich in other ways. Hi, welcome, Julie. Hello. How big is the class you teach? I'm going to try to keep the doxables to a minimum, but the class that I'm teaching this semester is quite small, so we'll have a bit more of an intimate experience in the classroom, which there are pros and cons to that, I think, where if it were me, I would actually prefer being in a bigger class because that means I could more easily be lost in the background because I tend to not want to stand out too much. That's just who I am as a person. But there are benefits, like the fact that you don't have to compete for my attention all that much. There aren't that many of them this time around. All right. Okay, so it's Friday night, and this is also a special stream in the sense that I finally learned how to raid on YouTube, which is not straightforward or easily accessible. So if you stick around to the very end, please stay on and don't click off as we raid into another science VTuber if she's still streaming by the end for me. School was a nightmare for me? Oh no, I'm really sorry to hear that. I know school can be hit or miss, and a lot of the experience can be determined by your environment, but I'm glad that you're here now, and we get to talk a little bit about biology. All right, Biology Trivia Night. Thank you to all of those of you who submitted a question. I can already tell that this is going to hurt my brand image as a biologist. Look, there's a lot of biology out there to learn, okay? It's impossible for me to keep all of the information in my tiny little nucleus, and so I'm hedging for when I get more than you might expect wrong, because I did take a quick peek at your questions, and they are not easy, or mostly they are not easy. So it's you against me, chat. I think last stream, I believe we actually tied, so the score's 0-0 in terms of victories. So we'll see how you all fare today. All right. So let's just get right into it. Biology Trivia Night, starting with the early. Oh, hopefully none of you saw that. In any case, this is the easy question. So question 1 is, what is the distinguishing factor between prokaryotes and eukaryotes? So this is a very basic question. I'll go ahead and start that poll. So, we're starting nice and easy. I think it was Thomas who had asked in my community post what defines easy, medium, and hard. I think of easy as intro level biology in high school. So I can't really speak to the quality of biology education in middle school, but I do remember high school. You know, I am of that age where some of my middle school memories have sort of faded. So introduction to biology, high school biology, that's quote unquote easy, although not all of them might be easy, because actually I cracked into an AP biology textbook and I was like, actually, this is kind of hard. It's actually kind of surprising how much they managed to cram into your head for AP biology. So easy is merely a marker of when you may have learned this information versus how hard it is to recall. All right, looks like we have a good bit of consensus in the chat right now. We've converged on D as an answer, so yeah. I kind of used the scroll wheel on my mouse by accident and maybe gave it away a little bit, but the defining feature that distinguished between something like a prokaryote, including archaea and bacteria, and eukaryotes is the lack of a membrane-bound organelle. So the things like answer A, the ability to cause infection, that's something that some bacteria have, not all. There are eukaryotes that can't exist as single-celled organisms, so that can't be the answer either. And the usage of binary fission to divide is not necessarily unique to bacteria, we'll say. All right. Good job, chat. That was a nice warm-up question. Certainly not easy if you are, or if you've missed a lot in biology. All right. Do cancer cells count as single-celled organisms? That's a very philosophical question that allows me to plug the repeated theme of my channel is that biology is a spectrum and categories are just things that we use to try to understand phenomenon. So I'll say definitely maybe. All right. Question 2. This is one of the harder quote unquote easy questions. So this is also something that you may have come across in your high school days, but maybe isn't that easy to recall. So again, easy is just a metric of when you should have learned this. Which of the following is not a body axis? Anterior, posterior, ventral, or retroid? Oh no, anatomy. I know. Anatomy is also my weakness. If I didn't do studies, if I didn't do research in the brain, I would have no idea. Because you could go through all of cell and molecular biology without caring too much about the body axes. But it looks like we have a pretty good level of consensus in chat right here. Where we drop in welcome, Clockwork, welcome. We are still in the easy round, so some of the gimme points, and we've got some good consensus going on in chat about the body axes. So I'm not a great anatomist, which is why you won't see too many anatomy questions in the questions that I give. But this is some basic biology stuff. All right, we've got some consensus, and it looks like most of you have said D, retroid. So that is something that I definitely made up. So let's take a look at the body axes. So I like to think of fish. It's just the easiest way for me to remember what the axes are because I always get dorsal and ventral confused unless I'm thinking about a fish. So we know from, early animal stuff, if you were a kid and you enjoyed reading about animals, and you would know that fish have a dorsal fin. So you just kind of have to remember that dorsal is like the back, quote unquote, of the organism. Posterior, we have, I think of the posterior as the butt because sometimes in English people refer to the butt as the posterior. So that one's pretty easy. Then anterior, I just think of that as the opposite of posterior. There's not really a great way for me to remember that. So then the opposite of dorsal will be ventral, and then retroid is just something that I made up. I don't know why I can't vote. I closed it when I noticed the consensus, so I may have just finished the poll before you got a chance to vote. Maybe you'll make it in the next one. Yeah. Does retroid stand for anything other than my parents mispronouncing Metroid? No, I actually spent like a solid 5 minutes trying to make up a word that sounded vaguely scientific, and that's where that's what I came up with. Dorsal ventral is easy to remember because in French it sounds like dos, body's back, and ventre, belly. Oh, interesting. No such comparison exists in English as far as I can tell, but that is neat. Also, welcome, Guy G. Welcome to the stream. All right, next question. This will be the last of the easy questions for you, and then we get into some of the more interesting stuff. So this is another tough question that is only easy because this should have been covered in introductory biology, but it is very tricky. So I'm going to go ahead and I'm going to start another poll. So which of these factors does not influence the frequency by which an allele for a gene appears in a population? So an allele is a version of a gene. A version of a gene. So say you have, in a very simplified manner, the blue eye allele and the brown eye allele, even though that's not how eye color works, but just pretend. The blue version of the gene is called the blue allele, and then the brown eye gene is the brown version of that allele. So allele just refers to variation in a certain gene. And I know that I've designed a good question when I'm seeing chat torn at exactly 50-50. Do we have some tiebreakers? It stands as a complete 50-50 tie. A trick question, if I'm not mistaken. It is very tricky. This is very tricky. This is a very tricky question. So we're still tied. 15 votes, we're still tied. There are 25 of you. You could swing it one way or another. Ooh, 17. We're back at 50-50. Oh, that's such a good question. I love this question. I love this question. Was never that good with genetics, SMH. I'm not great at genetics either, honestly. I struggled so much with the math involved in genetics. It was really, really tough. Actually, Clockwork and I were talking about just like molecular genetics evaporating from our brains when we don't need it. And it's true. I have much respect for geneticists, but it's just really tough for me to keep all of that in mind. All right. My question is, what even is genetic draft and is it real? I'm not going to say too much about that, but I will say that we are very, very, very slightly converging on an answer. It's so hard to answer these without knowing biology-specific terms in English. Oh, I know, I'm so sorry. This is the language that I was raised in, and so it is the language that I'm choosing to go with. And I have so much respect for researchers that come from different countries where they speak different languages, because if they want to come to the US to study, they have to conform to our dumb language. And English is in fact a very dumb language. So much respect if you can not only come to the US to study, but also learn all of our terminology in English. Just crazy. All right, so it looks like chat has settled on A as an answer. A. That's so close, right? A by 50%, D by 44%. Oh, this is such a good question. It allows me to talk a little bit about the nuances of genetics. So what is the answer? Did chat get this right? And I will say, so far, chat, you've done a great job. Dominance does not affect the frequency by which an allele for a gene appears in a population. And I remember having such a hard time in class understanding why that was, because you would think that, like, oh, if you get the dominant version of a gene, then you should see that more often. You should see dominance more often. But actually, it doesn't really matter, because dominance and recessiveness, that only affects whether or not you see the effect of a gene. It doesn't really change the chance that it gets passed on to the next generation, because that's influenced by the next three things, which I will go through one at a time. So natural selection, you've heard me talk about this multiple times. If you had content, if you've had sort of connection with my content in other means, so I've done shorts on evolution, I've mentioned evolution in a lot of my videos, so this should be review. This is the idea that the environment, or in Darwin's time, it was competition, but I think now we kind of see it just as an all-encompassing circumstance that a species finds themselves in. That context by which a species exists in will select for traits. So you can imagine that a bird with a very long sort of beak would be very bad at eating very tiny seeds. So the chance that a long-beaked bird gets to pass off the long-beaked genes in an island where the only food source are tiny seeds, that's going to be quite low. So natural selection absolutely affects whether or not a gene appears in a population. Then we have genetic drift. So genetic drift is the influence of random chance. So random chance is something that's just going to happen when you have something like a mathematically defined system. And genetic draft is a tricky term that I think should just be called genetic linkage, because genetic linkage is the term that I learned in class, but actually genetic draft is the, I think, the Wikipedia page. So maybe people are referring to it as genetic draft, but it might be genetic draft for a very specific reason, because it's looking at genetic linkage in a slightly different context. All right, so what are these? What are these? So genetic drift, this is the mathematical chance. This is just the random chance element. And the way that it is commonly explained is with this sort of random marble example. So imagine that every generation of a species is represented by a jar, and it's got marbles in it. And in this jar of marbles, we have red marbles and blue marbles. We'll pretend that these are two different alleles or two different versions of genes. And there is a random chance that any of these marbles will be passed off to the next jar. And so these dots represent the hit that the bead gets to be pulled into the next jar. So you can see that this bead was chosen, this bead was not chosen, this bead was chosen, this bead was not chosen. And so if you just randomly choose red versus blue, you could end up in a situation where the next generation has fewer red and more blue, and then over time you see the complete elimination of the red marbles, and this can just happen through random chance. So that can affect whether or not a gene is seen. So genetic drift can reduce genetic diversity in the absence of selection. So even though it doesn't really matter which version of the gene you get, red versus blue, it doesn't make a huge difference. Just random chance can sway your population and eventually decrease that genetic diversity. All right, so what is genetic draft? So the reason why it's called genetic draft is this phenomenon right here. So I don't know how many of you are into racing sports, but what you find in common amongst these racing sports is that people who are falling behind will want to race behind, like directly behind another racer. And the reason why this is because the racer in front of them will sort of tank air resistance for them and create this kind of vacuum, not a vacuum, like a void area where air resistance isn't as high. So you can see in cycling lines that, you know, each racer that is behind the other racer is experiencing less of that air resistance. And so genetic draft is kind of this idea that if you are following another gene very close by, sorry, no, that was fumbled. Say you're a gene and you happen to be next to a gene that is very important and is heavily selected for. Well, in that case, you will benefit by being attached to that very important gene. So how does that make any sense, or why would that even matter? This should be sort of review from high school, so I won't go into it that deeply, but during meiosis, not mitosis, but meiosis, which is the process by which your gametes develop, the cells that become your sperm and become your eggs, your chromosomes go under a process called recombination. So the data that you get from dad and the data that you get from mom will be sort of scrambled in a process called crossing over. So in black, we have a chromatid donated to you by your father, and in white, we have a chromatid donated to you by your mother. And what can happen is that a piece of that chromosome can then be swapped. So this one's from dad, this one's from mom, but in your sperm/egg, you get sort of these pieces of your genes that have been scrambled a little bit. Okay, so this is how it works. And so why would genetic draft change the frequency of a certain gene? Well, let's make this diagram. I colored it purple and orange. Let's say the orange chromosome came from mom. Let's say the purple chromosome came from dad. And let's say that there's this really important gene in green. And this is the gene getting selected for. Let's say that on an island, that there's a bird that has a tiny beak, and the island is full of tiny seeds. So there's a lot of pressure for that bird to pass on the gene for a tiny beak, right? But let's say there's a hitchhiker gene that's really close to the tiny beak gene. Let's say this gene is bird eye color, right? Something that you wouldn't really expect has any importance to the bird's survival. Just bird eye color. Maybe they're both two neutral tones. Like maybe the bird's eye is like brownish versus grayish, nothing huge that would give it away from a predator. So what happens during crossing over? Well, parts of the chromosome will rearrange. Part of mom's chromosome will rearrange with part of dad's chromosome. And this can happen in several different locations, and the location by which this happens is somewhat random. So let's say you have a crossing over event happening here all the way to the left here, right? If mom's chromosome crosses over with dad's chromosome here, then this is how this splits. And you'll see that if you get crossing over here, that the hitchhiker gene will stay attached to dad's chromosome, which has been replaced with a chunk of mom's chromosome. And similarly, if the crossing over event happens here, you still get hitchhiker gene getting separated with the gene that's important. And likewise on the other side, if you have the crossing over event all the way over here on the right, you'll see that mom's genes remains the same here, and then mom gets a piece from dad over here, but the hitchhiker gene and the gene that's getting selected for are still being inherited on the same chromatid. The only way for these two genes to get separated is if there's a crossing over event in between the chromatids. So, if this happens, where the crossing over event happens right in between, in this sweet spot between something not important like eye color for this hypothetical bird and something that is important for that bird, only in that case will a hitchhiker gene be able to be not inherited. Because we know that selective pressure will keep this gene in the gene pool. Does that make sense? Is that cool? The idea of genetic draft. Genetics is such a deep and rich field that I really think that like high school biology makes it seem too simple. And it's fine that high school biology is simple, but I think what is not more commonly done, or at least it wasn't done in my class, is that they teach you the simple version of genetics without letting you know that it gets way more complicated. So I remember getting my mind blown here. I wasn't ready for how messy genetics can be. I think freckles are a hitchhiker gene to red hair too. That's certainly possible. I'm not exactly sure genetically what the mechanism behind freckles actually are, but if that's the case, that would make sense. How often do these crossovers happen? So I believe I looked this up and someone can fact check me, but I think per chromosome, you get usually 1 to 2 crossing over events. Crossing over is also the way that we actually used to map genes on the chromosomes. And that might be fun for like a history of science kind of stream. Tracking the crossover events actually helped us determine which genes are where on the chromosome before we had fancier technology like sequencing, which is very cool. All right, I love this idea of genetic draft. I think it's very, very interesting. Good job, Chad. You've gotten all of the easy category questions right, so you all have a pretty solid grasp on your high school biology. So where do we go next? We go to the medium question. Now we're getting into college level cell and molecular biology. So definitely stuff that you should know if you are a biology major, and then stuff that you might be familiar with if you aren't. So here on out, it's going to get harder. Although, I know my audience. I think you guys will totally do very well here. All right, let's set up the answers here. Bring on the pain. Oh, Clockwork, I'm sure you'll be fine. I'd be really surprised if you got any of these wrong. So if you don't know Clockwork, he is my collaborator, or one-time collaborator, although I'm sure that we will collaborate again in the future. He does really cool biology, like protein biology videos that are very well animated. And if you like my content and you like the molecular aspect and you really want to dive deep, please go check out Clockwork's channel. Welcome, Silly Cat Boy. Welcome, welcome. So, what is the most common form of protein modification? Is it acetylation, ubiquitination, phosphorylation, or glycosylation? And as expected, we do have more of an uneven split in chat, which makes sense because this is a harder question that you might not know the answer to unless you've taken some serious biology classes. So we know from the central dogma that we go from DNA to RNA to protein, but what's not often as much talked about is proteins get modified, that they're not fully baked once they are made. They have to be finished, garnished, we'll say. Little protein garnishes. All right. some good chats, some good stuff in the chats. All right. Is there one more vote that we can get and make it a nice easy round number? Otherwise, I'm going to cut off the answers there as chat has converged. So we are somewhat split here. The most common answer is C. So what is the answer? It is C, phosphorylation. Good job, chat. So post-translational modification. So the process by which proteins are made is called translation, where you go from mRNA, messenger RNA, made into protein. But then after it gets translated into protein, you still have to finish it, garnish it with all sorts of different things for the protein to be functional. So these modifications can allow proteins to have a wide variety of different functions. You can ascribe new properties to proteins, give them new jobs to proteins, depending on what modifications are added on top. All right. So phosphorylation is the most common way that a protein is modified. And phosphorylation, if you're a biology student, you will see it all over the place. You will get sick and tired of phosphorylation. It is simply everywhere. Phosphorylation can do many things, but they are most associated with turning enzymes on and off. So up here in this top panel, we have, I believe this is serine. I'm not entirely sure. I think this is serine. And you add a PO4 group on top of it, and it changes very slightly the structure of this amino acid. And this might be enough to block its entry to some important active site on an enzyme or a protein. So by changing the shape of this protein, you've altered its function. And so phosphorylation is the most common way to do that. So Clockwork has a question. Ubiquitination is a secret trick answer because don't most proteins get ubiquitin tagged before they get recycled? I actually don't know if that is true. And I'm not saying that you're wrong, Clockwork. I'm just unsure if that applies to all proteins or if there are non-proteasome dependent mechanisms of protein degradation. I mean there are like the lysosome, right? So the lysosome is also a compartment that can recycle proteins. I don't know that it's always the proteasome, although someone can definitely fact check me on that. That's a great question though. N-terminal acetylation and glycosylation are more common. No, not to my knowledge, although I can maybe give chat another free point if someone can link a source that shows otherwise. I believe it is phosphorylation. All right, let us continue. Yeah, let's continue. Okay. Q5, what do the following proteins have in common? This is another inspired trick question. So followers of my channel, this one's for you because this is something I've been thinking a lot about recently. And I've talked about this kind of topic for a while because it's been a pet interest of mine. So what do the following proteins have in common? The first one being the major mammalian prion protein, something that I've talked a lot about. 2 is synuclein. A protein that I've not talked a lot about, but if you are interested in neuroscience, that's something that you might be familiar with. P53, which, I'll talk about the function of P53, but that's another very hot topic that any biologist will have learned about before they graduate. And 4, the snare complex, another very biology major heavy kind of thing. So this one's really tricky. So A, these proteins all aggregate and cause disease. B, these proteins are all primarily associated with the nervous system. C, these proteins are all intrinsically disordered. Or D, the function of these proteins are largely unknown. This one's a little tricky, so I'll give chat a little more time to think about what that answer is. So what do these proteins have in common? Let's see what chat thinks. All right. We are converging, but there is some disagreement. Can we get a couple more votes? Maybe try to even things out if there's disagreement. Please try not to spoil what people are answering in the chat. Let's just go in sort of naively here. Right, 22 votes, that's pretty good. I'll give you a second to finish up here. Ooh, we have a tie now. Anybody brave enough to be the tiebreaker? Anybody brave enough to be the tiebreaker? There are a couple of you that haven't voted, but maybe we should have it end in a tie. But if it's a tie, then you by default don't get it right, so that's so tragic. Still, there is a tie. Ooh, come on, chat. Come on, there's a tie. I'll give you 10 seconds, otherwise I'm ending the poll. 10, 9, 8, 7, 6, 5, 4, 3, 2, and 1. All right, we have a tie, which means, chat, you can't get this right because it is a tie, but I will say that the correct answer is in there. It is in there. So let's go over what these are. All right, so major mammalian prion protein. This has all of these as properties. So the mammalian major prion protein aggregates and causes diseases. These proteins are primarily associated with the nervous system. That's definitely true. These proteins are intrinsically disordered. Yep, the prion protein is disordered, and the function of this protein is largely unknown. That is true. There's still a lot of research going on to try to figure out exactly what this thing is doing. Not a lot about it is known. Synuclein. So this is much like the mammalian prion protein, a protein that aggregates and causes disease. So instead of, prion diseases like mad cow, this protein is associated with Parkinson's disease. That's a neurodegenerative disease that affects motor coordination, amongst other things. This is also an intrinsically disordered protein, as you can tell from this long, flexible tail that I'm partially covering. And the function of this protein is also largely unknown. Not really sure what it does. So these two fit the bill in all of these properties. What about P53? So P53 is gosh, I'm blanking on the word. It is it's a gene that makes a protein product, P53, that is important for apoptosis, which regulates cancer. So if you have a mutation in the P53 gene where it is not functional, you can develop cancer. In fact, this is one of the most common, if not the most common, gene that is broken in cancer. I believe 50% of cancers actually have this as a risk factor in them. So, this can certainly aggregate and cause disease. In fact, if these floppy aggregation-prone domains start to bind to one another and stick to one another, it can prevent P53 from doing its job, which is to tell the cell to blow up, right? Cancer is a disease in which cells lose the ability to regulate when they're supposed to remove themselves. And so by aggregating and stopping apoptosis from happening, it permits cancer to start to form. And you can tell all of these crazy spaghetti flops over here does mean that this protein is also intrinsically disordered. tumor suppressor. Thank you so much. Thank you so much. Oncogenic is the word that I was thinking, but tumor suppressor triggered the term in my head, oncogene, right? Oncogenetics, the study of the genes of cancer. But P53 is very, 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 very rigorously studied. We know a lot more about P53 than we do about synuclein or MMPP. Wouldn't P53 also be associated with the nervous system? So I said primarily associated with the nervous system, and P53 is not, primarily associated with the nervous system. And then finally, the snare complex. So the snare complex, for those of you who don't know, are a protein complex that helps cells with vesicle fusion. And I'm going to talk about why that's important. But for the snare complex, the only thing that applies to it is that its proteins are all intrinsically disordered. So C was the correct answer, although I purposefully made this one really, really tricky. So chat, not a perfect score this time, but I'm going to need that leeway because I think I'm going to bomb the section that you provided for me. So I'm glad that I'm not fighting an impossible battle, just a very highly unlikely battle for me to win. So vesicle fusion is really important. This is the way in which cells can sort of receive little packets of information from other cells. Most commonly, we think of the nervous system, but the snare complex is not only in the nervous system. It's not even primarily associated with the nervous system. It's just that neurotransmission is the most obvious application of vesicle fusion. So if you didn't know, the way that your neurons talk to one another is that they send little packets of neurotransmitters like dopamine and glutamate, and then those packets release things that bind to the other neuron, right? So how is it a protein that is how is a protein intrinsically disordered? It has an important function. It only causes diseases if it aggregates or quits doing its job. That's a good question. So let me answer that quickly because it's something that I realized that I never really addressed. Why would a protein be intrinsically disordered? Why would it evolve to do that? So I actually want to take a second here to ask chat that, because that is a good learning moment in terms of thinking about or developing a sort of imagination for biology. So why would a protein evolve to have disorder in its protein structure? I want to hear what chat kind of hypothesizes before we move on, because that is an interesting question. Why would a protein evolve to have floppy spaghetti parts? Like, why would it even enable the possibility of aggregation? Why even have this? Because evolution is pretty efficient, not always, but it's generally pretty good at making things that you need to survive. So with all of these proteins, except the snare complex, having this aggregation prone property, why would it bother? Why aren't all of our proteins super rigid? All right, some good questions coming in, good answers coming in. Intrinsic disorder just means it has a floppy side chain that sticks in the membrane. Not always. Not all of these proteins are membrane proteins. Or they don't have to be. A whisk, or maybe it needs to attach to things very flexibly. Yeah, flexibility, we are getting somewhere. Membrane tethering, certainly there's an aspect to that for flexibility. That is what I am thinking. Temperature resistance, kind of depends. That's not necessarily always true, but it can be. Potentially to originally save other, I'm not entirely sure what you mean by that. So if a cell is under stress, it wouldn't function normally if it's infected, for example. Not entirely sure what you mean by that either. Maybe the disorder helps with environmental changes, perhaps. Maybe it started as a change for it to be disordered to save other members of its proteins. Now it has survived despite that disorder. Not entirely sure. Cell movement, we're getting further. Flexibility is definitely where my mind is heading. So Imagine you need your protein to act as a sort of adapter molecule, a protein that can link other proteins together, something that needs to have flexibility so that it can fold in a way that can bind different things in different contexts. So say that there's an adapter molecule that wants to connect protein A to protein B. Well, in order to do that, it has to adapt a certain shape. It has to fit both protein A and protein B. And maybe there's an advantage that when A and B are not around, that floppy protein can then bind protein C and bring that closer to protein D. So this flexibility enables something like an adapter molecule to have different shapes at different contexts that will modulate what its function actually is. So that flexibility can really allow it to be more adaptive in its function, which is interesting. Faster reactions and more consistent interactions with their agonists. Not always, although that is in the right mindset in terms of reactions and interactions, I'd say. One gene, different functions, basically. Yeah, exactly. One protein, different forms. So that is a potential reason why you are able to do that, basically. Okay, all right. So let's talk about vesicle fusion. So vesicle fusion is a way for I hope that explanation made sense, right? If you have floppy spaghetti, sometimes that floppy spaghetti will do X function when it's shaped like X, but then you can like refold it into a different origami shape and then it has Y function. So that sort of flexibility can be quite adaptive. Whether or not that flexibility is important for the function of the prion protein or synuclein, that's not as obvious, although there's a decent amount of flexibility with things like p53 and other transcription factors. Sorry, I shouldn't say transcription factors. I don't actually remember how p53 works, but there are certain transcription factors that need to be flexible so that it can bind flexibly to a whole bunch of different genes, for example. Okay. Where was I? I kind of got sidetracked. Right, vesicle fusion. So there will be instances, for example, say the lysosome fusing with the phagosome. If you've seen my tuberculosis video, that's one example of vesicles needing to fuse. And one way that vesicle fusion happens is through the snare complex. And I love the snare complex because there's something very satisfying about solving this impossible problem, right? And I think everybody will have their own angle talking about snare. I was actually talking to Clockwork behind the scenes about how cool the snare complex is. And actually, when I pitched the idea of talking about snare, we had completely different ideas of what makes them cool. So if Clockwork ever publishes a thing about snare, I will let you know, because I can never stop hearing about how cool this thing is. So the vesicles in your body are made of compounds called lipids. So lipids are like little fatty molecules that comprise of cell membranes as well as these little pockets called vesicles. And lipids have this chemical property. They are a fatty material, and they have a negatively charged head and a hydrophobic tail. So if we look at this diagram up here, we see that this round piece, this loves water, and then these tails, they hate water. Hello, background character. Welcome to the stream. Talking about vesicle fusion as a part of my trivia stream. And so here's the problem, chat. Vesicle fusion, energetically, from a physical point of view, is an impossible problem, because this head has a negative charge, and so when you bring two vesicles together, you are bringing two things that, A, they love water, so they really want to have water bound to them. But not only that, you should know from middle school that like charges repel. So if you bring a positively charged probe next to a positively charged probe, you'll get repulsion. And you would know this if you've ever played with magnets, that if you try to push the negative side of a magnet to the negative side of another magnet, that they repel. That's this, but at a much smaller scale, right? The negative charge on one vesicle and the negative charge on a membrane, those negative charges will repel. So how does this cell solve this problem? Right? This problem of electrostatic repulsion, this idea that these negative charges attached to the membranes, if you try to just smash them together, they're not going to want to combine because they have all these negative charges. So chat, if you know the answer and if you know what snare is, try not to ruin the surprise. Let's give the non-biologists a way to ideate and appreciate the beauty of nature. So, you, the non-biologists in chat, how would you design a mechanical system that would be able to merge these two layers? Don't worry, I've removed your comment, Clockwork, and there's not a lot that you can get out of your comment anyway, but just to have the non-biologist in the most pure state of ideation. You have two surfaces, two electrical surfaces, two negative charged magnets. How do you get them to play nice and actually fuse? And please, if you're a non-biologist, let your imagination go wild. My guess is that both ends grab onto each other like an ISS dock. I'm actually not sure. Oh, like the International Space Station? Is that what ISS refers to? That's kind of interesting. Make a hole in both membranes so that they can connect. That's an interesting idea as well. That's that, and I'll say that that's interesting for a different reason. That's not correct, but it is a part of the story. Something that pierces both membranes so that it can break them apart and reattach them to each other. That is going in line with what Guy G is hypothesizing, and that is, again, that's part of the story. And I actually really like that because it is kind of correct, but you want something first has to happen. You first have to bring them together. So you're both, Thamor and Guy, you're both correct, but not in the right time frame. Nucleating opening. Yeah. Maybe there's a protein in the membrane that does that. It can. There's a complex called the snare complex that'll bring these close enough for what Guy and what Thamor and what Marlin are saying. You're all correct, but we need to go back like 5 nanoseconds before that happens. Bring one into another. How? E2, you've identified the part that the snare complex is doing. It's bringing and smashing these two membranes together so that they can fuse. But how? A fishing reel. Okay, a fishing reel. Okay, now we're getting somewhere. A fishing reel. Anybody have something like that they can think of? What else could bring them together? Emit another magnet in front of the two opposite poles when they attract. That would be something. Does it have positive charges on the outside that latch onto the surface of the vesicles? Oh, that's interesting. I don't know the exact answer. I believe that is accurate, but not the answer that I am looking for. I like fishing reel. Can we go down that line of thought? What other things could bring these two together to force them to interact? Because right now the electrostatic repulsion is pushing them apart and we need some kind of mechanism to bring them in. A fishing reel could definitely do it. But what else? A black hole. I mean, certainly, although the vesicles maybe would not survive that process. You need to attach two close membranes together so the process of fusion starts. Exactly. You need to have them squeeze them hard enough together. But what actually can squeeze them? A clamp. Yeah. They have a big get along shirt. Yeah, that would do it. Not the answer I'm looking for, but I do love that answer. Some sort of unstructured protein that acts like a spring once it's anchored. Ooh, that's interesting. Like a spring, an unstructured protein that acts like a spring once it's anchored. We're getting closer. Maybe something with a cytoskeleton. That is a part of the story, but a part of the story that I'm not really focused on right now. It's using fibers as scissors. So that part, not necessarily. But I like where we're going. So we're going to combine the idea of a fishing rod with this idea of a spring and anchoring, and we are going to get the snare complex. And so this is super cool. So it's not quite a fishing rod and it's not quite a spring, but you'll see how those ideas kind of explain this. So On the surface of the membrane and on the surface of the vesicle that is to be fused are the proteins of the snare complex. And so what happens is that these three proteins, syntaxin, SNAP25, and synaptobrevin in this particular vesicle, they have these floppy spaghetti-like domains that just flop around. They're kind of like Velcro. And so once a membrane vesicle gets close enough, these three proteins will start to tangle in a triple helical bundle like a zipper. Like a zipper. So because it is like a zipper, you start to decrease the space in which, as you zipper in and tighten those bundles, kind of like what, oh, I forgot who said it earlier in chat, this idea of a spring once it's anchored, like this spring that anchors these membranes together. And once those zippers sort of bundle up tight enough to reduce the space going from these unstructured hairs that are just flying around to this really tight triple helical bundle, you get an arrangement where you bring those membranes, you overcome their electrostatic repulsion, and then you can sort of force them to combine, which I really, really think is very, very cool. Oh, did I make a JoJo reference? Or did someone else make a JoJo reference? I must have missed it. Would you be willing to explain the near impossible part after this? I could have sworn Capra says it does happen spontaneously. Oh, so I'm not like an energetics person. Like, I'm not really a thermodynamics expert. And I do believe that technically Thamior is correct where anything can happen with enough chances, although not anything. But I mean, there's a chance that you happen to have the energy to overcome the electrostatic barrier that exists between membranes, right? So there's this barrier, and there is a chance that you smash them hard enough naturally for this to happen spontaneously. But I believe for the most part, there's usually some sort of mechanism by which this has to happen. Things like micelles, which only have one layer, maybe that's an exception. And also it depends on the geometry of the vesicles as well. So it's not, so sorry, I don't know enough information to completely address that situation. What I will say, what I do know off the top of my head is that the geometry of the vesicle, so their curvature, can certainly influence the chance that they spontaneously combine. But when you learn about vesicle fusion, the snare complex is often brought up as something that helps remove this barrier, this energetic barrier that exists between like and like charges. I hope that is a satisfying enough answer. So you can think of a vesicle with a very severe curvature, like a very tiny curvature. You can see that the flat surface of electric repulsion is less, right? So the more severe that curvature is, the less likely you'll have a large surface area for mutual repulsion. And I believe off the top of my head that that could potentially explain some of that away, although I'm not 100% certain. Ah, zippers. Yeah. Sticky fingers. That makes sense. Sticky fingers. Oh, God, I'm out of it. Yeah, no, absolutely JoJo reference. I meant it all the entire time. Botox, I think, makes a cut in the zipper helix and causes the whole thing to unzip. Oh, interesting. I'm actually unsure of how Botox works on a molecular level. That'd be interesting to look into in terms of why it causes paralysis. All right, so I love the snare complex. I love this idea of proteins overcoming energetic barriers. It just makes for a good story. And your ideation in chat, that ideation is something that I want to cultivate, especially if you are a younger student, because that kind of ideation is important if you want to go into engineering. And so I'm a big proponent of that. Wikipedia says SNAP25 makes some cuts in. Oh, that's interesting. I actually didn't know that that was a part of its mechanism. I was always taught that the snare complex was mostly there to bring the two vesicles close enough for fusion to happen. Maybe I'd be really curious as to when that research came out. Either I forgot or maybe it's quite recent, but that's interesting. That like nicks the membrane. You made it sound way more interesting than when I first learned this. Yeah, that's kind of my idea of the channel, is that like I want to bring you the part of biology that I appreciate. I only want to talk about biology in a way, and Clockwork and I have talked about this too, like we won't talk about stuff unless we have the right angle. So snare is something that I definitely think has an interesting angle. And I do owe a lot of this to my professors at my university because they impressed upon me the beauty of some of these systems. Oh, Botox snips the SNAP25. Oh, and maybe that prevents vesicle fusion from happening and shuts down communication between neurons. That would make sense if I were to hypothesize. But okay, so chat, that's one question down for you. So that's 1 out of 9 that you've gotten wrong, but you're on pace for a very, very good grade. And now let's move on. So question 6, this is the last of the medium difficulty questions. Let's start that poll. So this is a topic that I am paying respect to, despite me not being very, I'm not an expert in this field, but I do acknowledge that there are a lot of people who are very passionate about this topic. Which of the following is not a nucleic acid secondary structure? A, double helix, B, reef knot, C, stem loop, and D, pseudoknot. And I am actually going to be right back as I get a drink of water as you go ahead and answer that. All right, I'm back, chat. I have my water. I also want to sort of impress upon you all this idea that, like, I think of my role as a science communicator as being sort of like a museum guide or something, like an art appreciator. I would certainly teach this very differently if I had you all in my class. It's just in this space, I get to be an appreciator of science. And so that's what I think I'm doing is much like how I was I had like mandatory art and music appreciation classes in high school. But here I get to have a science appreciation class. And so that is my angle here. Oh, chat, we're caught. Oh no, this is a majority. Okay. I think I'll probably cut it here because this one's a bit more of an esoteric question. So what did chat say? Reef knot, B. So let's see what the answer is. So this is what a double helix looks like. I'm sure you should be familiar with this because that is the principal secondary structure that DNA takes. There are different forms of DNA, different forms of the double helix, but DNA pretty much just has this going for it. The stem loop in C looks like this, where RNA, because the backbone is much more flexible, you get to have all these fun different shapes. So this one's called a stem loop. And this one is called a pseudoknot. And so I am not like an RNA guy, but RNA guys are very passionate about RNA. They're very like super hardcore from my experience, because RNA are interesting biomolecular materials. DNA is sort of exciting in its own ways. You can build origami out of DNA, but you can also build all sorts of wild different shapes to RNA, which allows it to have properties that make it very interesting for an engineer or a synthetic biologist. Ribozyme moments, exactly. Like, unlike DNA, RNA can contribute to catalysis or chemical reactions, and part of why it can do that is it can adapt all sorts of very interesting, powerful shapes. And a reef knot is this. So, good job, chat. You have identified the knot that I kind of just threw in here for fun. Yeah, there's certain RNA sequences that incorporate into the ribosome. Absolutely correct. RNA is so interesting and very cool, but I am less of an expert in that. Is the stem loop the same one in tRNA? I believe so. But it's been a long time since I thought about the biology of tRNA, but I believe that is correct. Somebody can correct me if I am wrong. Okay, chat, you have made it to the end of the medium round with just one mistake. And now we get into the hard stuff. So the hard stuff, I am defining as stuff that you would generally not learn as a part of your undergraduate education. So that doesn't necessarily mean that these are harder. It just generally means that you would not likely have this information in your 101 Intro to Biology class as an undergraduate. Like, it's not covered in AP Bio, not covered in undergraduate biology. This is some more specialty stuff. You would have to have taken a class specifically on bacteria, I think, to get this question correct. So, and I've talked about some of these before in streams and stuff. So this is maybe not the hardest question, but it's a nice warm-up to this category. So which of the following is the phenomenon by which bacteria can survive antibiotic treatment largely independent of inheritance? Is it A, bacterial persistence, B, antibiotic resistance, C, bacterial stasis, or D, bacterial survivorship? This one's tough. Oh, chat's going crazy. Oh, we are neck and neck with all sorts of different answers. Yeah, this one is really tough. This one is tough. All right, any last takers? We've got a tie. Got a tie between two answers here. Yeah, looks like we're tied. Very, very interesting. Oh, okay. We're getting up there. All right, answers are still coming in. Okay. All right, I think I will end it there. And we'll see what the answer is. Okay, so chat, you answered B, antibiotic resistance. So the answer is A. Unfortunate for Thamior, it is as literal as it actually is. We name things for a reason, generally. This is actually not referencing spores. This is a very neutral mechanistic approach to a definition because this idea is somewhat mysterious. The largely independent of inheritance makes me doubt my answer. Yeah. I learned about persister cells in my microbiology class, my bacteriology education coming in clutch. Exactly. So very relevant in biofilms. Exactly. So let's learn about what each of these actually are, because I think I did my rotation in a lab that studied persistence as a graduate student, and I was obsessed with this idea. I think it's so cool. So here's antibiotic resistance. It's the thing that most people learn about for good reason, because antibiotic resistance is not quickly becoming a problem, but is definitely currently a large and increasing problem in terms of our never-ending war against the pathogens that want us dead. So in antibiotic resistance, you have a colony or a population of bacteria, and you treat them with an antibiotic, and most of them die. Let's say that 1 in a million bacteria happen to have a gene that shuts down that antibiotic, like a penicillin or ampicillin resistance gene. If you take that survivor and you allow that survivor to multiply via culturing, so you take away the antibiotic and allow it to grow happily in, I shouldn't say that. If you treat that bacteria with antibiotic and it lives and you continue to culture that bacteria, you'll see that that population grows back up. But if you take that same culture, this large population of bacteria that have had an antibiotic resistance gene, and you treat that population with antibiotics, they all survive. This is what we commonly known as antibiotic resistance. For a given bacterial population, there is a non-zero chance that there's one or several of those individual bacteria that have something in their gene, in their genes, in their DNA, that allows them to repel or neutralize certain antibiotics. And if the only survivors have that gene and those bacteria get to continue to grow, they will form a new population that are all resistant to that antibiotic. So what is persistence? What makes persistence different? Say you have this population of bacteria and you give them the antibiotic, and that leaves just one surviving bacteria. If you take away that antibiotic and reculture that bacteria, you get the expansion, you get that bacteria happily growing. But then you give that antibiotic again, and it seems like the bacteria didn't genetically remember how to deal with that antibiotic. So hopefully, chat, you can see why persistence is a different phenomenon than resistance. And the other two answers I just totally made up. The stasis answer is a bit misleading because if you'll see this is very neutral. This is an observation. This term is named after an observation so that it is mechanism neutral. And so I may have fooled some of you with bacterial stasis because as some of the chatters are coming out and saying, some of bacterial persistence can be attributed to biofilm formation, right? So if there is an antibiotic challenge, some bacteria will try to survive by forming a protective biofilm. And that's not something that is genetically encoded in them. It's just, we're stressed, we're dying, we need to survive, so we're going to make something that allows us to continue to survive. But that might not be the only mechanism that is at play in bacterial persistence. And we're learning more every day about all the different ways that bacteria can overcome antibiotics, not just through an inherited gene, but by other means, including biofilms, but not limited to biofilms. So I hope that's a little interesting for you to chew on, this idea of persistence. I was obsessed with this bacterial communication and hardiness because bacteria are pretty incredible. Pseudomonas moments, exactly. Pseudomonas being one of the bacteria that you go to when you study biofilms. Could a biofilm colony count as a multicellular organism beyond a certain point? Bacteria in a biofilm don't always have to be in direct contact with each other. And again, this goes back to what is biology? What are category labels? They're just ways that we try to label things so that we can understand and describe them. But since they don't necessarily have to make membrane contact, it's a bit harder of an argument to make. All right. Next question. So this one's for the neuroscientists in chat. So that was, I believe, 2 questions wrong so far. So this is the next of the hard pool questions. And this was mainly an excuse for me to show some very pretty immunofluorescent data. So the red cells here were stained with an antibody against IBA1 in a slice of the rat cerebellum. So I don't expect any of you, unless you have neuroscience background, to get this correct. And so you should be able to, if you study the brain, be able to pick out what cell type this is by the marker, which is IBA1, and the morphology, so the shape of the red here. In blue, we have the nuclei of cells, and in the red, in the green, I'm not entirely sure what they stand for there, but the red is what we care about. So what's in red here? Very pretty picture. I'm so cooked. Yeah, I'd expect you to. If you're focusing on bacteria, there's no reason why you would know this. That's why these are hard pool questions. Just an excuse for me to talk about cool biology that I like. All right. The cerebellum is a great cue to remove one answer. Yeah, I didn't want to keep it a secret. We're looking in the brain, the cerebellum. So what do we think, Chad? Joke's on you, I studied histology. Drat. I specifically picked an immunofluorescent one because histology tends to not use immunofluorescence. And I don't actually know if the markers are the same between histology and immunofluorescence, because I've never done histology in my life. But dang, I think icosahedrons got this. The med school OP advantage. All right, I'm going to call it there again because this is a bit of an esoteric question to answer. So we'll end the poll there. So chat said B, astrocyte. So the answer here is actually D, the microglia. So IBA1 is a marker for the microglia. The microglia is the resident phagocyte of the brain. So the equivalent, you could kind of think of them as the equivalent of the neutrophil, although that's not really correct. But the microglia are a supporting cast member to neurons in the brain. They help like recycle debris in the brain if you've had injury or infection. But they also help support the neurons as they grow. They help regulate the neurons and help them signal. And for those of you who have watched some of my other material, you would know that they also have a role in pruning synapses, which I think is really interesting. They chew up sort of the extra connections that you don't need in your brain so that you can make the connections in your brain more efficient. So these are microglia. I was so surprised I guessed right. I started choking. Yeah. Yeah. This one's tough. Omim said it was expressed in immune cells. Yeah, microglia. Again, with like labels and categories, I like to just refer to them as resident phagocytes, because to call them immune cells, they certainly do look like immune cells, but to give them a more like neutral term, I think phagocyte is a more appropriate name because that more describes their function without the baggage of calling them immune system cells. I saw the radial star pattern and thought astrocyte. Yeah, but astrocytes can get quite big, which is why this was like a morphology kind of question. So relative to the nucleus, an astrocyte would be much larger, although it's not a bad guess because astrocytes do have a similar pattern, but microglia are much smaller, hence microglia. I'm pretty sure it was B because the shape was incompatible with A and C. Not always. Neurons in culture, if this was a cultured dish, can look kind of like this early on in their development. If we're talking about like a 3-day-old neuron, it might not look that different from microglia. All right. So I've gotten a little bit of wiggle room to at least tie with chat. So that's very helpful. So that was... So it's the persistence question. So I've got 3 questions of wiggle room on the upcoming part. Okay, that's good. And number 9. So again, there's no reason why you would get this if you don't have advanced biology knowledge. So, don't be afraid. Although also don't be afraid to guess because guessing is fun. Which of the following techniques allow you to deduce the identity of a protein without the use of antibodies? Without the use of antibodies. So if you've watched my content, you'd be able to at least eliminate one of these answers, even if you knew nothing about biology. Which of the following techniques allows you to deduce the identity of a protein without antibody? Ooh, we've got a lot of agreement going on here, but not all the votes are in, so there's still time for chat to be split. What do we think, chat? All right, we're tinkering, but we are converging. Okay, we are converging, and I think we're not going to get a huge swing after this many votes are in, so we'll call it there. So chat answered C, mass spectrometry. So we'll see what the answer is. So A, Western blot. If you've looked at my shorts, you would know that this is not the answer. I've done a short on the Western blot, so you know that that's not true. You definitely need antibodies for that. And ELISA is a technique similar to a Western blot, but instead of relying on things like immunofluorescence or radiography, you have a colorimetric readout that you can do in a 96-well plate. There are pros and cons to the ELISA and the Western blot that I won't get into details here, but they both serve a similar but not identical function. The mass spectrometry. So this is the correct answer here. And the mass spectrometer is so cool. Let me just explain to you in just geek out terms, I should call it, because this is such an incredible piece of technology. So we know that proteins are made out of amino acid building blocks, right? And so what the mass spectrometer can allow us to do is to identify proteins by ionization of their building blocks. So each amino acid will have a different mass to charge ratio. So isoleucine and leucine, they're slightly different in size. They've got slightly different molecules as a part of their structure. And the charge between their mass and charge, the ratio between their mass and charge are slightly different. So what you can do is you can take a whole bunch of chewed up protein and you can ionize them, make them into plasma and shoot them through a field of magnets that will bend their location, their projection into a detector that will correspond to their mass charge ratio. It's freaking sci-fi, dude. Like the idea to take advantage of the fact that there is a unique mass charge ratio for every amino acid, and then to, I don't know, like for me, instrumentation people are incredibly like godlike in their genius, right? This is such a cool idea to take advantage of this property, shoot them through plasma, and then use magnets to sort of bend them according to their mass charge ratio is insane. So if you have a protein sample, you can actually chew it up and then spit them out into a plasma torch. And then by the way that they bend through the magnetic field, you can actually reconstruct their sequences and identify those proteins. Absolutely insane. Just not lasered proteins. Yeah, exactly. Mass spec is based. I know. I love mass spec. Having worked with one though, dude, this machine is always broken. This machine is always broken. For those of you who are thinking about a career in biology, if you want to make bank, if you want to make money, and you like biology and you like machines and like are natural, have natural tendency towards engineering, be a mass spec technician. Oh my god, the money you can make by unclogging these godly machines is incredible. They say there's no money in biology. Well, if you have a background in instrumentation, there is money in biology, believe me. Multi-TOF is broken, understandable, have a nice day. Exactly. My first rotation was in a lab that had a mass spec, and I never even got to use it. And I was also babied the entire time, because if I clogged it, it would have meant misery for the rest of the lab. So I was watched like a hawk. Very cool. If you want to make money in biology, go be a mass spec technician. Okay, D, the Bradford assay. So the Bradford assay is a colorimetric assay that is used to quantify proteins. It has nothing to do with determining their identity. So that's what that one is. All right, I have a wiggle room of 3 questions to get wrong in this bonus section. So I won't poll chat. You can keep, record for yourself how many you get right here, but now it's the chat against me. So chat got 3 wrong. How many will I get wrong? These are your biology questions. So last trivia night, I had asked for general science questions. This time, I wanted to raise the stakes. If I get any of these wrong, it will look bad for my brand. So I'm very invested into not screwing these up. I've not looked at the answers, obviously, but I have looked at the questions beforehand so that I could prepare these slides. So that's just full disclosure. So it's not completely fair. I did have a little more time to think about them, but I promise you I didn't really think about them too much. All right, a note on submitted questions. I didn't include questions with too many joke answers. If it was obvious that you weren't genuinely trying to stump me, then I didn't include them in there, sorry. And I didn't include questions that were already covered in other trivia streams. So if your question isn't here, either I didn't get to it, in which case you have priority in the next stream, or I didn't put it in because it's only fun when it's tough, right? Trivia is only fun when you kind of have to struggle a little bit. All right, let's get going. Let's humiliate myself. Okay, this one's from Eason, toddler tosser in the Discord. Which one is wrong about the plant family of... I don't even know how to say this, dude. I saw this question and I was like, oh my gosh, I am cooked. For those of you who have followed me for a while, you know that plant stuff, if you want to just guarantee that I get an answer wrong, or a 75% chance I get an answer wrong, ask me about plants. I'm really bad at plant biology. So I'm going at this purely from the angle of psychology, trying to suss out what the answer is likely to be. So every pair of leaves are rotated 9 degrees like a whirl. Sounds like something that could be right. You probably ate some last night is a very fun answer that I think is also correct. I don't know how often two different genuses or two different species names or family names are redundant with one another. So this one was on my radar, this B one. And then quadrangular stems. Quadrangular just strikes me as a fun, jokey name. I don't know if that's an actual word, but my answer here is going to be A, quadrangular stems, just because, I don't know, the rest seem very plausible. All right, I'm going to scroll down. The answer is B. Okay, so the explanation that Toddler Tosser gave is Linderniaceae is a different family related to Lamiaceae. Jesus. The answer can be found by looking at the name Ca, which indicates that it's at the level of family. Lamiaceae, also known as mint, includes catnip, lavender, and herbs like basil, sage, oregano, rosemary, thyme, and mint. Probably the first family of plants to be identified for someone new to plant taxonomy like me. That's so cool that you're studying plant taxonomy. Not cool that the first question is a question that I've gotten wrong. I am truly shivering in my timbers right now. Oh no. Okay, so I have 2 more questions I can get wrong before I tie with chat. Latin problems. Yeah, I know. This one's really tough. So that answer is B. Okay, so I'm 1 down, 2 more to get wrong. This one's from Anonymous. What is the mitochondria? I won't count this. But, like, I think this is acceptable, but for the spirit of the game, I have an extra question at the end here. Powerhouse of the cell, we all know that. This is not going to stop me, and so don't worry. I have thank you for the submission anyway, but like to make up for this difficulty, I did add another question at the end of this presentation, but I just wanted to acknowledge that the submission was received. Okay, so question 12, Marlon, I don't know if you're still in chat, but this one is your question. What is the name for the principal type of glial cell in the peripheral nervous system? And I thought, oh god, I am so cooked, because my research was primarily based in the central nervous system. So thinking about the peripheral nervous system, like the nerves that go out and innervate the organs and the muscle, I thought, oh god, I wasn't even like aware there were glial cells in the peripheral nervous system. So I am purely going off of elimination and just brute force recognition here. So I've never heard of a Miller cell, so I'm going to say that that's not it. Mueller, I think M with umlaut U, I have heard of, although I don't really know. Schwann cells are supporting cells that help make up the myelin sheath in neurons. I don't know if they're glial. I don't really know that that's true, and I don't know if that is a distinction between peripheral and central, so I'm gonna say not Schwann. An astrocyte, I believe, is primarily a central nervous system thing. But that's only because I recognize astrocytes in the central nervous system. This is a tough one. Mueller I've heard of, but I don't remember in what context. Oh, it's gonna look so bad if I get this wrong. I want to say Mueller cells are like in the kidney or something. Oh god, this is so. That's why these are so. This is a really good question because this is really high stakes for me. I don't know that Schwann counts as glial. I say that out of pure ignorance. Like they might be. I just don't know that. So, oh gosh. Yeah, it's been too long since I thought about the peripheral nervous system. I think astrocytes are primarily central nervous system, so I'm going to actually go... Because there are Schwann cells in the central nervous system too. Ah! This is agony. But I'm going to go with B. Mueller. I'm gonna go with B. Mueller. Oh gosh, this is gonna be really bad if I get this wrong. Okay. Let's see what the answer was. I'm waiting for the answer to load here. The answer, oh, no, that's not the one. That's the mitochondria one. The answer here, Schwann cells. So Schwann cells are the primary form of glial cells in the peripheral nervous system, coming in from myelinating and non-myelinating varieties. The myelinating variety gives peripheral nerves their characteristic myelin sheath. That, yeah, I got that part right. Important for efficient axon potential progression. Oh, so they do count as glial. I didn't work in my background wasn't in myelination, and so I didn't have to learn. Like, so for context, when you're in grad schools can vary quite differently in terms of how many classes they make you take, and so a lot of the information you just kind of pick up as you need it, and I never really thought about myelination all that much. So the flaw here for me was I didn't know that Schwann cells were technically glial, and not some other categorization of cells, because my main focus was in neurons as neurons and the stuff that neurons do as neurons instead of this accessory, even though I should have, which is why this is such a good question, is because I should have known the answer, but I didn't. What the heck is a Mueller cell? Am I just making stuff up? Mueller are also glia. Oh, they're in the retina. Oh, I knew them from somewhere. They're in the retina. See, that's my central nervous system bias. Dang. Good question, Marlon. Really stumped me. Maybe I wrote the question bad. No. I think it's really me overthinking the question and not technically knowing what Schwann cells counted as, because I was not very focused on glia when I was in graduate school. That's good. Good question in here. There's a small trap in this question. The equivalent of astrocytes in the PNS are satellite cells. Schwann cells are the oligodendrocyte cousins. Oh, interesting. I mean, I still would have gotten this wrong anyway. Because I picked the thing that's actually in the retina. Tough. Dang. Yeah, Schwann cells of oligodendrocyte. Yeah, interesting. Oh, yeah. Good question. I'm very happy to take that L, and also very, very unhappy to take that L, but that's what makes it so good. So I only have one more room for error question here. You have all been kicking my butt. So this one's from icosahedron, and this one is mean too. How many cells compose of a blastocyst, a 1-week-old embryo? So when I looked at the numerical options, I thought, oh, okay, I'll just pick the one that's a power of 2. So a blastocyte. So for those of you who don't know, sperm enters the egg and forms the zygote. Hopefully you've gotten that from high school biology. And then the embryo starts dividing in powers of 2. So this is not necessarily a universal mechanism of embryonic development. There are some cases in which some of the cells will actually apoptose and then you don't get powers of 2. So I thought, okay, for humans, I know that is powers of 2. So I was like, oh, I'll just pick the answer that's a power of 2. And icosahedron, oh, you made them all powers of 2. Oh, you scoundrel. Okay, so this is actually quite tough for me. A week-old embryo. So the blastocyst, so this is before gastrulation. Oh god, I had to memorize this as an undergrad. Yeah, you would know this if you took embryonic development recently, but I haven't. Blastocyst. So I'm like, I've got the image in my head, and I don't think it's 256. I think that's too much. I don't think it's 32. So I'm between 128 and 64. I think 64 sounded like an important stage. Like I remember hearing 64 a lot in embryonic development, but it could also be 128. I'm going to say, because I recognize the number 64 more, that a blastocyst is at 64. I thought this was easy and hard at the same time, since you could just remember this randomly. Yeah, I mean, it's another really clever and dastardly question. I'm gonna say 64. I'm locking in my answer as 64. 128. Damn. 7 divisions to 128. I was so close. I knew it had to be 128 or 64. It's not looking too good for me, chat. I've tied with chat. I can no longer get any more wrong, or I will lose to chat in this round of trivia. 64 is indeed important. Do you know off the top of your head what the 64 stage is important for? Because that's the one I remember learning about. For some reason, that's the thing that sticks ahead in my head. There's some other important checkpoint at 64 that I don't remember. But dang, that's a good one. That's a good quiz. See, this is the type of question that I love to get for these trivia streams, because I'm supposed to know this and I don't. And that's what makes that just a really good question to ask me and humiliate me with. Okay, next question. This one is from Olhaldo179. Thank you for the question submission. So let's get a look at what this one is. How does mycelium communicate to the rest of fungi as fast as it does? So I know very little about fungi. So this one is cool. But the idea of communication between fungi being very quick, I want to say that the answer is A. Because hormonal cascades, they can also potentially be very quick, but I think there's a hint in the question that it's got to be fast. So while hormonal cascades can be relatively quick relative to other forms of communication, we don't typically think of hormonal communication as being fast. So I'm going to say A, electricity similar to a neuron's action potentials. How do I do the poll? Is it just for members or something? So this section is actually just for me. The poll was actually for the questions that I came up with, but these are chat submitted questions. And this one's just for me. You can keep track on your own as a personal counter to see if you beat me. But this half of the trivia night is just for me so that I get roasted. Just makes things a little more smooth. So I'm saying that the answer is A, electricity similar to a neuron's action potential. Oh no. Okay, so the answer that Olhaldo gave was we don't know because fungi are weird little guys. Oh, no. I have already lost to chat. You have bested me. Good job, chat. I've taken the L on this one, so that's made I think I may have won the first one, although I'm not sure. But I think we are at least tied overall in these trivia streams. Wow, yeah, so if you're an up-and-coming biologist, maybe you want to get into studying mycology, because certainly there's a science prize of some sort if you can probably decipher how fungi communicate. That's so interesting. Oh, by the way, just for the record, for posterity's sake, I haven't fact-checked any of these submitted by chat, so I'm just trusting that all of you are correct. There was that funny mushroom robot article making rounds. Yeah, I saw that too. That's why I really thought that they were able to communicate through electricity. Because how else would they be able to make that robot? That's so interesting. Day 5 mark is when embryo takes its spherical form. In the period from 64 to 128, the cells outside and inside specialize. So when the germ layers are determined, question mark? Mesoderm, ectoderm, and then the other derm. Maybe that's what I was focused on. My take for Q14 was transfer of molecules through gap junction, so something similar to electrical synapses. Yeah, I would have imagined that that's a pretty sensible way for something to evolve to have communicated. Mesoderm, endoderm, ectoderm. Thank you very much. Yeah, so maybe the 64 number was when I was thinking of mesoderm, ectoderm, and endoderm. Oh, dang. Taking big L's. Okay, this next question's from Thomas. What genetic principle allows for all 4 basic blood types, A, B, and O, to exist with only 3 alleles? So codominance, incomplete dominance, epistasis, genetic linkage. So this one's another great question that I should know the answer to, especially because I kind of covered this in my genetics lecture. But as I have repeatedly mentioned, I'm not a great molecular geneticist. So I believe that there are several different definitions or several different descriptors that can apply to the inheritance of blood type or the expression of the blood type alleles. And so I believe what this one is going to get at is codominance, because you can have A and B expressed at the same time. How do you vote? This half is just for me. This one is just for me. It's chat against me. I'm the only one going to get roasted here, although you can keep track on your own personally how many of these you have gotten right. Okay, so I'm going to say codominance with regards to blood types. So we'll see what Thomas says. Codominance. Oh, thank God. That was going to be really bad if I got that wrong. Codominance is when two alleles are both expressed at the same time, like in individuals with blood type AB, which have A and B alleles, so they can produce both antigens in their blood. Incomplete dominance is when neither of the two alleles are dominant, resulting in a blend of both traits. Epistasis refers to one gene modifying the expression of another, and gene linkage is when two genes are close enough to each other on the chromosome, so they are often inherited together. Yeah, so we saw gene linkage earlier in my section, a question that I had asked. Hello from Iceland. Welcome to the stream. Welcome, Mikhail. Are blood types caused by human leukocyte antigen HLA, HLB, HLA-C expression or independent of that? I actually don't remember. That's another good trivia question that I'm supposed to know because I studied HLAs a while ago. Good questions in chat. Good questions in chat. All right, next one. This one's from Bacilli Boy, Bacillus anthracis. Don't know how you say that. So Bacilli Boy is a VTuber who streams about biology on Twitch, actually. You can go ahead and check them out. They have a sugar named after them called Anthros, Bacillus, or Antos. So it makes less sense for a sugar to be named after the genus. So I'm going to say that Bacillus is likely not the answer. So that's going to be between Anthros and Antos for me. And for me, Anthros just sounds more right. It sounds more like a sugar than Antos for some reason, although that's completely a vibe-based check. So, Bacilli Boy, my answer to this is going to be A, Anthros. Anthros, okay, great. It is the terminal sugar on the oligosaccharides on the exosporum of Bacillus anthracis spores. In 2020, there was a paper looking at anthros. It's called Convergent Evolution of Diverse Bacillus anthracis. I'm also not sure if I'm saying that right. Outbreak strains towards altered surface oligosaccharides that modulate anthrax pathogenesis. Oh, so this is anthrax. I guess that makes sense. By Michael H. Norris et al. Involving anthros. Okay, so this might be a subtle hint to me that maybe I should cover this in some sort of phi content. Interesting. Yeah. I mean, in terms of like the stuff that people want me to cover on my channel, I'm gonna get to all of the things that you want me to cover. I'll cover Black Plague, I'll cover brain-eating amoeba, all in good time. It's just, my fear is that if I cover all the eye-catching stuff that you want me to cover, maybe anthrax included, that by the time I'm done with them, you'll all leave me for some other biology content creator. So I want to stretch out those very eye-catching videos with stuff that I genuinely love about biology to kind of hook you, to get you hooked on biology before I give you those more widely requested videos. Very spicy sugar. Apparently, yeah. Great question. Great question. All right, next one, question 17 by Panda, the parietal epithelial cell. The splitting of what complement system proteins start the membrane attack complex leading to the lysis of pathogens. So I love the membrane attack complex, but I am what I call a fake immunologist. I never took a single immunology class, even though I studied immunology in grad school, which is why, if you are an undergraduate, I highly, 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 highly recommend that you go broad and not go deep in your undergraduate education if you want to achieve higher education. Right? If you just want to graduate with a bachelor's and go work, then going deep is fine. But if you want upper level, if you want master's or PhD or postdoc professor level aspirations, I encourage you to go broad, because in graduate school, nobody's going to make you take extra classes, right? In graduate school, you just pick up what you appear to need. And I didn't have a formal education in immunology. Everything I know about immunology, I picked up on my own. So That being said, which of these complex proteins is the one that splits? So I think it's C5. And the only reason why I think it's C5 is because I recognize C5 and I don't recognize the others. That's the only thing. So this is another case where I might just randomly know the answer, but I'm not entirely sure. Okay, so Panda, the answer that Panda provided was C5, thank God. C5 splits into C5A and C5B, notably C5B through the C5 convertase, and that starts the membrane attack complex. So the membrane attack complex is a super, super cool immunological tool that can bust open bacterial cells. They have this like Autobot, like transformer protein complex that all comes together and pops bacteria. And part of that cascade is the splitting of the C5 protein, which again I just randomly recalled because C5, I just remember C5 being really important. Nice. If you get an answer wrong, you initiate an immune response in an inappropriate situation and cause an autoimmune condition in its host. Yeah. Oh, my bad. Great question, Panda, great question. All right, next. So this one's the last question. So I gave you 9 and then chat gave me 9, or I picked 9. This one's from Tony. What was the first antiviral treatment? Lysis mean popping, I assume. Yes. It doesn't actually pop. A lot of lysis people get kind of annoyed when people say that. It's also apoptosis instead of apoptosis. So the idea that cells pop when they lyse or when they pop when they undergo apoptosis, that's just factually wrong, but I casually refer to it as pop because what is true is that the bacteria can no longer keep its insides on the inside and its outsides on the outside. So that's why I like to think of popping as a way that happens. Okay, so first antiviral treatment, antibody-producing bacteria. I don't think that one is correct because I don't know. I just don't think if we could have bacteria make antibodies, we could have vegan vaccines, but or if they can make antibodies, they might not be very good at making antibodies. Or if they make antibodies, maybe that's not good for the bacteria. I imagine bacteria making immune proteins, they might not be very happy with doing that. So I think A is not correct. Wait, is it not actually apoptosis? My world has been shattered. Yeah, you can find a lot of annoying apoptosis people out there who will insist that we keep close to the Greek root of the word apoptosis. I don't remember what the root actually means, but they insist that it's apoptosis with the soft P. Interferons for SB pick, they are immune signaling molecules that alert the immune system that an infection is occurring. Virophages, so these are viruses that... Virophages. So does that technically refer to phage that co-infect other phage? I don't think that would be an antiviral treatment. Bacteriophage maybe, but I don't think virophage would be correct. I understand that it's not true, but I enjoy the theatrics of a cell popping. Exactly, I know, it's so much more fun to think of them that way. But if you want to be picky about being correct, which is valid on my channel, I was picky about Plague Inc. and all of my Plague Inc. shorts, so it's valid to be picky on my channel. I totally accept that. So interferons, I think that's probably going to be correct. I don't think in terms of the first antiviral that we would have gone with something as dangerous as a bone marrow transplant. So I'll say interferon. I also fail to see how a bone marrow transplant can be an antiviral. So let's say C, interferon. All right, Tony, what is the answer? Interferon. Okay, great. Interferon treatments were first started in the 50s and are the first antiviral treatment. Yeah, all right. Interferon. So that allows the immune system, it signals to the immune system that there's an infection occurring, and it is activating the immune system to fight off infection. So the difference between antibacterials and antivirals is actually a really fascinating distinction that you don't often think about because they work in different ways. In bacteria, there's a lot of antibacterial compounds that do things like blow up bacteria, not technically blow up, but pop them the way that I'm colloquially referring to as popping them. Destruction type stuff. But in terms of antivirals, they're more focused on preventing those viruses from replicating, and pumping up the interferon response could be a way to help the immune system do more of that. Yeah. Apparently, as a biologist, you have to be correct, huge if true. I mean, as a content creator who's constantly being lightly roasted in my comment section about getting things technically 100% right, I'm used to this idea of like, I need to be as right as humanly possible, or the comments are going to bubble at me. All right. So, to replace the mitochondria question, I will go with one more question here. So this one's from Anonymous. Which one of these medications can cause vision loss? So we've got an annoying medicine person in chat. I'm sorry, I love you medical folk in my chat. I love you too. But medicine, it's a lot of knowing things. I already have enough on my plate with biology. Okay, so let's try to do some tactical process of elimination. So amoxicillin, that is an antibiotic. I don't think that would have vision loss as a side effect, although there's no reason why it wouldn't. It's just, since amoxicillin, the primary purpose is to stop bacteria, that doesn't seem like it would have an effect on something in the nervous system. Chloroquine, so this one is, I'm not looking at chat. This one is malaria. So malaria is an infection by Plasmodium falciparum and other species in the Plasmodium genus, which is they are eukaryotes. Amoeba? Are they amoeba? Ooh, I should really know that. I think they're amoeba, but it's another anti-infection, we'll say. Wellbutrin. So this is a name that I recognize, and I don't remember why. If I recognize it, it might be a nervous system thing. Metformin, I don't recognize at all. I recognize Wellbutrin, and I just feel like if I recognize Wellbutrin and I don't know where it is, then it might be a nervous system thing. And if it's a nervous system thing, maybe it's a vision loss thing too, because the eyes are heavily connected with the nervous system. So I'm gonna say Wellbutrin. This is a great question. So, Anonymous, what is the answer here? No way, Jose. The answer here is chloroquine. So chloroquine can accumulate in the eyeballs, causing chloroquine retinopathy. It is a drug used to treat malaria. Okay, so I was right on the malaria thing. I was wrong on it not having an effect on the eyeballs. Again, like, there's no reason why an antibacterial can't cause vision loss. I just didn't think that it was likely. Okay, so why do I know what Wellbutrin is? What is Wellbutrin? What is Wellbutrin? Oh, okay, so it is a nervous system thing. So Wellbutrin is an antidepressant. Okay, so I was like, why do I know that this is like a nervous system thing? It's an antidepressant. Okay, wow, okay. So at least I kind of remembered why I knew Wellbutrin, but it had nothing to do with the right answer. Oh gosh. Dang. I would love to know what percent of people have that side effect. I know, that's so unique. I definitely didn't know that. Let's see, I'm gonna catch up on chat because I was really pondering. Yeah, chloroquine is for malaria. Yep. Antibiotic, anti-malaria, metabolism, question mark. I guess I'll also look up metformin just to round out what these drugs are, because I am kind of curious. Metformin. What is metformin? Metformin is for diabetes. Okay. Okay. Gotcha. Okay, so that's what the metabolism answer was, or a quote in chat was. Wellbutrin is an antidepressant. Yep. Shot in the dark. Okay, so geez Louise, you and I we were barking up the same tree, but we were both wrong, unfortunately. Wellbutrin is also named bupropion and is a dopaminergic, not really an antidepressant. Oh, I just kind of Googled and read the first thing that I read, so apologies if I'm propagating misinformation on the internet. Oh my god, I'm the king of guessing. That's an incredible guess if you got it right for the wrong reasons. Yeah, good job. All right, chat. You've bested me. You've bested me at my own game. You've given me biology questions that I could not answer, and your aggregate intelligence bested me as I tried to stump you with the questions that I submitted. So, I don't have a particular award for you all. Just, I'm proud of you. You've attended your biology classes. And again, I'm always surprised at how much biology knowledge you all have as an aggregate. Good job, and continue on with your studies. Those of you who have stayed, I'd love to know. Since you have all bested me, I wanted to kind of get a sense for where you guys are at in terms of your education. So this is not like how old you are, just like what is your last experience with biology. And also please stick around, we're going to raid into Faye if she's still streaming, because I finally figured out how YouTube rating works. But before we raid into her, I just kind of want to check university and then postgraduate. So I just want to check, because I'm always kind of curious, especially because you guys have kicked my butt, what your last experience with biology is. So whether or not you're still in high school, or whether or not your last biology class was a high school class, or whether or not your last biology class was a university, or if you have biology experience beyond undergraduate. Because I'm just kind of curious, who shows up to these streams. Starting biotechnology undergraduate in 3 weeks. That's very, very exciting. That's so cool. I hope some of these thought-provoking questions, especially the one on snares, gets you in the mindset of an engineer. I love that. I love that. I'm an engineer, but my dad is a neuroscientist and my mom is a doctor, and I'm genuinely curious. So you've sort of osmosed a lot of this information. Comp neuro PhD student. Comp neuro, my nemesis, Nick Garcia. In my department, we were one of the only molecular biology based labs in the neuroscience department. The rest of them were comp neuro people, and comp neuro, man, you all are really smart, but yeah, I feel like a lot of you forget that we exist too. So here's my plea to you, Nick. Please, for the biologists in your audience, make sure your presentations are accessible for them as well. Postgraduate in biochem. Very fun, very fun. I thought about doing biochem in graduate school, but at the graduate level, I found biochem to be too hard. You've read biology textbooks for fun, CS higher education. There's a lot of you out there who have CS background in my audience. I find that so fascinating. Third-year student in biochemistry and molecular biology. Heck yeah, man, heck yeah. I majored in molecular biology myself and really loved biochemistry, so I'm glad that you are here listening in. I'm a senior aerospace engineer in university. Last time I took biology class was in the 6th grade. Wow. I hope that my questions really appeal to the engineers, because these thought-provoking mechanism questions, you can take that as inspiration for whatever you engineer. There's a lot that comes out of biology that gets used in all sorts of different fields, so I hope that some of this stuff is useful. Microbiology undergraduate, very, very cool. No wonder you know all the microbiology questions. I shouldn't be surprised. So it looks like 50% roughly are university students, and the rest of you are split between high schoolers and university. So that's like a really good space for me to be. I love seeing that I have people currently in training. And I also, don't get me wrong, I love that we have some postgraduate people in the chat too. You can help keep me honest and fact check me. I really could use that, because it's always risky doing science communication and risking communicating something that maybe isn't super accurate. The rivalry is real. It is. Like, believe me, like I tried to go to your seminars, man. I tried to learn what you were all up to, but you just start talking about algorithms and I get lost so quickly. So I do, like, genuinely, molecular biologists do want to know what you're doing. We just need a little help, okay? Doing 12th grade bio right now as an adult because it's a prereq for university, I'm guessing. Interesting. I hope some of this can stimulate and keep it interesting. Biomimicry is really cool. Definitely. I think it's cool that your channel has attracted so many different people. I know, and I'm meeting more of you as I stream. Like, I love doing the videos. That's my primary artistic outlet, but getting to talk to you in streams and seeing not only the chat members that I recognize, but also newcomers to the stream. I love that, like, I'm getting to actually talk to you because I am a teacher by training, and my passion is to engage in an active manner, not just having you watch my content, but to hear from you as well has been a lot of fun. Biology nerd world domination. Heck yeah. In your opinion, what's the best way to prepare for uni? Just, there are a lot of different ways you can prepare for uni. You probably will be homesick, but I recommend that if you are like me, like a brutal introvert, just tell yourself for a week, just be the biggest extrovert you can be. Just force yourself to do it. Don't be like me and ignore all the orientation events because it's overwhelming. And not only should you be the biggest extrovert in your first week or two, you should also acknowledge the fact that you might not make friends in those weeks, but what's important is practicing the art of socializing with your peers who are unfamiliar. So you don't have like the baggage of high school and inheriting sort of experience from that. For the most part, people who go to university, especially if it's in a university in a different state or in a different country or a different area, it's really important to practice the art of attempting to make friends. So I was very lonely my freshman year because are you surprised? I'm a freaking VTuber, right? It's basically a meme at this point that every VTuber is this like omega introvert cooped up in the room all day, and that does describe me quite well. But I would recommend you force yourself to do that. And I forced myself to do that in graduate school. And then I fell back into old habits. But if there's one piece that I would say, be extroverted. It's only for a week. And also, the second you need help, go get it. I can't tell you in my teaching career, not saying in any particular timeline in my teaching career, but along my long teaching career, because I've been teaching in some form since I was, in high school, I cannot tell you the number of students that I've seen that will just suffer. They'll just suffer. They'll just bomb and they won't get any help. And in university, you're an adult. It's not my job to make sure that you graduate. It is your job to make sure that you perform. But it is my job to help you when you need it and when you get it. It is my job to be your help. But I cannot help you unless you seek it. So that, like, be an extrovert in your first 2 weeks and get help exactly when you need it. Don't wait until like you're forced to take a leave of absence from university. It's a waste of time. It's a waste of money. Get the therapy that your university likely pays for. Even if it's not good, try a couple therapists. Talk to your academic advisor. If they're not great, talk to other adults in the room. Like, just when you need help. I cannot stress this enough. That's such a good question to ask, because this is my plea for all of you undergraduates out there. If you start bombing your classes, please get help. Please get help. Please get help. Yeah, so that's my soapbox. If I had time, I would pop into more. I'm a TA for microbiology. Great, that's awesome. This semester, definitely going to plug your bacteriophage and prion videos because people always have so many questions on those. Thank you so much. Yeah, thank you so much. That's awesome. Have fun TAing for microbiology and doing all the staining and all the fun stuff. I love microbiology, even though I didn't specialize in that in graduate school. As you can tell, primarily what I communicate on this challenge is on this channel is microbiology. I just find the stories of the little guys so fascinating. Reading more into chat as I was ranting. Get into a club. Yes, icosahedron. Yes. Join a lot of clubs and quit a lot of clubs, right? Make sure that you can do well in your classes, but at the same time, don't be afraid to try a whole bunch of different clubs. This college was, you're never going to have an easier time. And I'm just speaking just straight reality, being hag age. That like college is the easiest place to make friends. It's not the only place. Adults can make friends too. Like it's not impossible, but if you want like making friends on easy mode, that's university. Making friends on hard mode, that's working adult life. So while you have that ease, try to make friends, and don't keep fake friends around just because you call them your friends, but really make the effort to meet a whole bunch of people. That's the best use of college. People say that college is for making for giving you a job. That's true to some degree, but the intangible benefit is the ability to be connected to so many different people. So make sure you take advantage of that. I literally watched you in Clockwork because you guys make it interesting. The course I'm taking is rough. There's a lot of struggle in biology. I have a short-term memory capacity of 5, which means if you tell me a telephone number, I won't remember it, which made studying biology very hard. But I have this kind of mentality where like your competence in biology is made up of not only your passion and your natural ability, but also your hard work. So if you find yourself struggling, the only thing that I can do is root for you. Biology is tough, and I personally had to put a lot more blood, sweat, and tears than my peers who had, photographic level memory. But I did it because I love it, because biology is great. Being an undergrad is a huge transition. It can be taxing on your mental state. Yeah, definitely. Just reach out for help when you need it. Great advice. I love that everybody is giving in their input, because college is such a potentially great time. That's why I say potentially, because I don't want to give you false hope or anything. College is what you make out of it. In the US, you pay a crap ton for it. A lot of people think you don't need it. I'll say, if you want a job in biology, you kind of need it. There are jobs in biology where you don't need higher education, but they don't pay very well. So if you find that your passion is academic, then of course college is going to be very useful. So I don't like this like attitude that people can have of like, oh, you don't need college to make money. Yes, that's true. You don't need college to make money. You need college to make money if you want a job that you need college for. And people forget to say that a lot. So just, keep that in mind as well. Freshmen tend not to struggle because of academics, but because of how much responsibility you have compared to high school. Yeah, this is like, I've been thinking about this a lot. Like, I feel like, and I don't know this for a fact, but like COVID, I think maybe was really tough on a lot of people in terms of being home all day and not having like separation from your parents. And like, it might be harder coming from a generation where you had just entire stretches of your life where you were with your parents 24/7 to like mature into someone who can handle life on their own. And I don't want to make assumptions about people. That's just a hypothesis that like, it's like maybe being home every day for 2 years like wasn't great for starting to transition. So I like acknowledge that it might be a little harder coming from COVID, but with that in mind, you still are expected to function at the level of a full adult in university, and that transition can be pretty rough. Oh, it's not that hard. It's just presented in a really boring way. Yeah, that's why I love this channel, because I get to just talk about stuff that I like. And as an artor, like as not an artor, but like as a connoisseur of biology, like I've always loved biology, and professors, they love biology too, otherwise they wouldn't be professors, but their love is in doing biology. My love isn't in doing biology, my love is in communicating it. So I feel like I can inhabit the space of someone who gets to just eat and savor biology, whereas they're the cooks in the kitchen. I think it's a really good analogy, actually. Your professors and professional researchers, they're in the kitchen, they're cooking up new biology for me to just nibble on, and then you read my review in the New York Times about, oh, this biology tasted really good, and then that's what I get to tell you. Academics are also hard. Learning how to study. Yeah. I was thinking of doing a how to study biology stream, but I think I might wait a little until most universities have already started, because there is academic research in the teaching literature, which I also occasionally peruse because I am a teacher. So I'm interested in teaching research about what are effective methods of studying biology in particular. And, people will say, take notes. Taking notes is fine, but it's not sufficient. So people who recommend taking notes as a way to get better grades have cracked memories to begin with. As someone with terrible memory, taking notes was absolutely terrible. It was not... I actually stopped taking notes because I found that taking notes for me, with my bad memory, was not useful. Research has shown that there are there's one founding principle in terms of increasing your grade, and that is the art of active learning. So I'll do a whole stream on this at some point, but you can look this up. The research in it is good. Classes with active learning components or force their students to actively learn during lecture, and that can include note-taking, but it's not always enough. If you are doing something to learn, that is active learning. If you are reading your notes to study for a test, that is not active learning. If you're just reading, and if you're just trying to memorize by reading, that's not enough. You need to actively learn. And the best way, in my opinion, and from the research that I've read, is to test yourself. It's annoying. It's really annoying to do that. But give yourself practice tests. Write questions that you think could be on the test and see if you know the answers. Simulate the testing environment. The closer the testing environment is to your studying environment, the less stressed you'll be, and the more likely you'll be able to recall the information, especially in biology. Taking notes help me focus on the lecture, but I don't use them afterwards. Exactly. Taking notes is an active form of learning, but reading notes is actually very passive and not very efficient in terms of studying. So I wouldn't recommend that. Don't let this scare you and other pre-college peeps. You'll find your way. Yeah, exactly. It's a big deal, but at the same time, you're going to be fine. But if you find yourself not being fine, go get help. Teacher interaction is huge for me. Any question the professor asks, class, I'm trying to answer. Yeah, and you don't have to raise your hand to answer. When your professor asks you a question in class, they're not really oh, so-and-so is really smart and I'm going to write them a letter of recommendation letter, which is certainly possible. But the teaching reason, the behind the scenes reason, is that by forcing yourself to answer a question like this trivia, it will actually help you recall that information because you are actively using your noggin to work. If you are just reading your notes, that's the worst study material. People who read their notes to study for tests have incredible memory capacity to begin with. You can rewrite your notes, that's better. Practice tests are the best in terms of, the amount that you can improve your grade, but if they don't have those, make your own. Yeah, the more active you can be in your learning process, the better. Okay. With that said, I am getting tired. I kind of want to see if Faye is still streaming because I really want to raid into her stream because she's a great VTuber. She's actually one of my personal recommended. If you want to watch a VTuber be really good at chatting, Faye is an incredible chatter, and she also does a lot of biology content. So please stay on the stream, and while the stream ends, it'll actually raid directly into Faye's stream. So with that being said, thank you all for showing up to my stream. Thanks for playing trivia with me. You all did great. Of course, you beat me. The school year is starting, so for those of you who are starting classes again, good luck, have fun, and I'll see you again in the future. Thank you all. Go raid into Faye and tell her, if you are going to be raiding into Faye, let's come up with a raid message real quick. I don't know. I think it'd be fun maybe just to say mitosis followed by 3 dots, right? So when we raid into Faye, if you find yourself redirected into her stream, just say mitosis with 3 dots. All right? All right. Bye, y'all. Goodbye.